If you're interested in thrillers and crime stories, we will give you some eye-opening facts today. Have you ever wondered how the police convicts a criminal only with small parts of fingernails or hair? Thanks to everyone's unique genetic fingerprint, perpetrators can be identified easily. But what defines your genetic fingerprint? And how can it be compared between different individuals? The technique you use for this analysis is called gel electrophoresis. With its help, protein chains, or in our case DNA sequences, can be separated by their individual size and charge. For the determination of the genetic fingerprint, repetitive regions in your genome are compared with so-called simple sequence repeats. The repeats are located in the exact same region in your genome. They only differ in the number of repeats and therefore in their length. Now, gel electrophoresis comes into play, because it is the main technique to compare the length of DNA sequences. The technique is based on a pore-forming gel similar to a net, to which voltage is applied. Due to the negative charge of DNA, the fragments migrate to the positively charged pole. Large DNA fragments are not able to move fast in the gel, the small pores hold them back. Precisely the opposite case is true for short DNA sequences. They are much more agile and therefore migrate further than long fragments. This is why the comparison of the unique repeat length by electrophoresis is a certain way to convict a perpetrator by its genetic fingerprint. However, gel electrophoresis is not only used for fingerprint analysis, but it is one of the most common methods in the biochemistry lab. Every step of genetic modification can be verified by measuring the size of certain DNA fragments, for example of a specific gene you wanted to transfer into your host organism. Amplifying the genetic sequence through PCR and comparing the sequence length to a standard marker gives information about the success of your experiment. This way, you can also isolate genes for further analysis or search for proteins with the right size in your sample.